Good evening, everybody. Welcome to the third version of the ECNP Daily, third day of the ECNP Congress in Barcelona. I'm sitting here. My name is Ilse Graat. I'm a member of the Early Career Advisory Panel. And today I have three guests again for the ECNP Daily. Sitting next to me is Roger Adan. He is a professor of molecular pharmacology at the University Medical Center Utrecht at the Utrecht University in the Netherlands. His research concerns unraveling the neural circuits that underlie behaviors relevant to psychiatry with a focus on eating disorders. He's also on the editorial board of Neuroscience Applied and on the scientific program committee for next year in Milan. And sitting next to him is Olivia O'Leary from Ireland. She's a senior lecturer at the Department of Anatomy and Neuroscience at the University College Cork. And her main interest is the neurobiology and treatment of stress-related psychiatric disorders with a particular focus on depression. And next to her is Annamaria Cattaneo from Italy. She's an assistant professor at the University of Milan, and she's also heading the Laboratory of Biological Psychiatry at the IRCCS Fate Bene Fratelli Institute. My Italian is not so good. She's particularly interested in evaluating how stress early in life, as well as exposure to depression in utero, can shape the brain trajectory versus an enhanced vulnerability status. Recently, she has also focused her attention on the gut microbiome as a link between the environment, immune system, and brain functionality. Thank you all for being here today.
Thank you, Ilse.
I hope you all had a great third day here at the Congress. And maybe to start with, you can share one key takeaway from the sessions that you attended today. Roger, do you want to start?
If I may start,
Yeah.
well, what today for me was very clear that translational models deliver. So I attended the new findings in applied neuroscience today, the S10 session, and there were young scientists who showed their work, and it was amazing that really these translational models have impact for diseases like Parkinson's and eating disorders. So that's really very good news. And if I may continue on this is, of course, a symposium where young scientists were selected because they visited the ECNP workshop in Nice. And every year, this is an initiative of ECNP that they select 100 young scientists to come to Nice, and they get lectures, and they present their work, and they get some training. And then we select people to come to this Congress because they performed so well. And of course, that's why it worked so nicely that we had this symposium today. And also, if I compare it with how I started with neuroscience 35 years ago, nowadays as a young scientist, how quickly you can incorporate molecular neuroscience technologies like fiber photometry and optogenetics in your work. It's really amazing and really a fun area to work.
Yes, so you were very impressed by these
I was impressed.
Early career.
I could redo my career and then start all over with the point that I'm now also at. But now it's really amazing what a fun it is to do neuroscience research nowadays.
And a really great workshop. I attended it myself a couple of years ago. It's it's amazing.
Myself as well years ago.
It's where you start. Exactly.
And I would encourage people to really make people alert. It's still possible this month to apply for that for that workshop. So encourage your PhD students and young investigators to prescribe, and maybe they get selected and get an opportunity to, in the end, also participate in this Congress.
Yes, at the New Findings Symposium, and yesterday also the ECNP Got Talent. Also really nice format.
It was really nice.
Yeah, great. Good highlight. And for you, Olivia?
Well, today I really enjoyed, we had educational session number 4 on ketamine and psychedelics and the potential that they may have for the treatment of depression in the future. because the problem of treatment-resistant depression and difficult-to-treat depression has been there for such a long time, and there has been really little hope, I suppose, that we may overcome that, and that this large proportion of individuals that are treatment-resistant would not have improved quality of life. But today we had an educational session with Professor Alan Young and Professor Klaus Norman, really highlighting some of the very interesting and promising work showing that ketamine may be, very useful for broad clinical use, and also highlighting the potential of psychedelic treatment as well. So I think that was really important, and we had a very nice discussion as well about the neurobiological underpinnings of this. And this really highlighted the importance of synaptic connections, those connections between neurons, and how maybe if we can accelerate those connections and the pruning of appropriate connections, that really may be the key to more effective antidepressant treatments. And we saw this again today, actually, in one of the symposiums, and with some very nice neuroimaging data presented by Gitta Moos-Knudsen, who was our former president or past president of ECNP, demonstrated very nice neuroimaging studies in humans, demonstrating changes at the level of the synapse in response to psilocybin, in response to ketamine. So we've now come from a situation where we're coming from preclinical studies, now translating into humans, and potentially back again to inform us with more mechanistic mechanisms.
So nice to hear in that area also.
Yeah.
So animal models really deliver, huh?
Mm-hmm.
And this goes maybe a bit against the public domain opinion, but it's really, and particularly for the field of neuroscience, I think that we will still need them also in the future.
Of course,
Despite, of course, the developments. Because I was just before this recording, I was attending the presentation by Sergei Pasca on human organoids, and it's amazing nowadays what we can already do with skin cells from patients, and then we can differentiate them and really make small organoids that resemble parts of the brain, and even making combinations of these organoids so that they make connections and so on, and you get an enormous diversity in cell types that resembles the human brain. And I think what we need in the future is kind of complementary studies. Of course, we need to see to what extent these new findings in a dish translate to the real situation for a brain intact in this physiological context.
And these will have huge implications for precision medicine, for precision psychiatry,
Sure, because they can even take a patient with a mutation and then with CRISPR-Cas correct it, and then see how that impacts on the physiology in a dish. Yeah.
So interesting new findings in those symposia.
Yes. Yeah.
And for you, Annamaria?
Actually, this morning I attended one of the industry sessions where they were emphasizing the importance of anhedonia to be targeted, because anhedonia is associated in depressed patients with social functioning, with suicide. So they emphasize the importance of try to target specifically anhedonia, also to make patients back to feel emotions. And so to try to, use specific antidepressant drugs that are able actually to improve specific domains in depression. And then also attended the lecture of John Cryan. He's always inspiring. So I really enjoy the lecture. He's because he's able to keep the attention from everyone, but at the same time to deliver important message. Yes. And one important message that I think it's really something that we need also to deliver to a young scientist is not just to stop at associations, because associations can be random, as he mentioned, but we need to go more into the detail about the causality, so which is the causal relationship between one specific behavior, for example, and a mechanism. Because in this way, if we do know which is the causal mechanism, then we can target then that mechanism in more targeted way. Yeah.
Yes.
Yeah, the way that the bugs talk to the brain was all correlational data, but
Yeah.
now they go beyond correlation, and we're really
Exactly.
seeing now
Yeah.
Yeah, of course, in
mechanisms.
humans, it's more
Yes.
correlations.
And I think it's
that's-
really important to unravel mechanism to convince that that interfering with the bacteria
Yeah.
has therapeutic efficacy in the future.
Yes.
Yeah.
Yeah, it was very interesting.
Yeah, and also another sessions, if I can, that really enjoyed is the inclusion in science. I was impressed, especially about the last sentence that also she wrote in the abstract. She was indeed saying that if you are smart and you put effort, then you don't need your hands. But if you are not smart and you don't put effort and you don't really put effort in what you are doing, then even hands are not useful to you. So, and this, I think it's a message that can be applied in general science and life. Yeah.
Yeah. And Annamaria, you're
Yeah.
also part of the abstract and poster committee.
Yeah.
Yeah. How did you see any of the posters today?
Yeah. So actually this year a lot of posters and abstract were received, more than 700.
Wow.
And I think it's really good because it means that ECNP has all the year is really highly appreciated by young scientists because it's a way during the poster presentation where they can explain what they are doing, their project, they can interact with the other, and of course also get inspired by other young researchers.
Yes.
Yeah.
And any poster that stood out for you today?
No, there were many posters.
Many.
Some of them of today received also an award, will receive an award, so yeah.
Yeah. Nice. Yeah.
So difficult
very-
choices to make.
It was a difficult choice. Indeed, actually,
quite.
the meeting lasted more than we planned because it was really difficult. Many really good posters were received.
Yeah, difficult. Yeah. And in your expert opinion, what are some of the emerging trends in neuroscience that you've seen here on the Congress?
Well, there was the lecture on Engrams was really inspiring to me.
Yes.
It turned out that if you have a memory trace, there are certain cells that get engaged, but it turns out that these are already pre-activated, and I think this means that these cells are not alike. So, and I can imagine that, for instance, in my field, hunger primes certain cells, and in a situation of hunger, certain cells get more engaged in learning than when you're satiated, for instance. So, and that explains why, for instance, receptors for ghrelin are also in the brain on these circuits. So they may engage certain cells in that process. I thought that was something that I really thought that is interesting. It's all based upon modern technologies such as optogenetics, vibrophotometry, and even that you go with a lens into the brain and see the activity of the cells while an animal is moving. And it's really amazing. We learn so much about how memory is formed, how it's stored, how it's retrieved. And I think this is going to have an impact on, for instance, Alzheimer's disease, because we need to understand, how memories are formed and processed and so on.
Yeah. So inspiring session
I found that really
to take
inspiring.
home as well.
Yeah.
Yeah. Yeah. And for you, Olivia?
I think I really enjoyed as well that there's a little bit more emphasis now on, I suppose, the impacts on the whole body and the brain in different brain disorders. So we're not really talking just about the brain, but what happens in the body affects the brain, what happens in the brain affects the body. So, for example, we had a very interesting campfire session yesterday about vagus nerve stimulation. And the vagus nerve is a very interesting nerve because it's the only cranial nerve really that goes beyond the head and the neck area, and it innervates lots of our internal organs. So it's very well positioned to relay signals from lots of places in the body to the brain, and for the brain to communicate with the rest of the body. And this particular nerve, we know, has, there's a tool called vagus nerve stimulation. It's invasive, where an electrode has to be placed around the vagus nerve, the level of the neck. But it has shown efficacy in treatment-resistant depression. But now there is kind of a move towards maybe trying to find a more non-invasive approach to that, where you could have a transcutaneous stimulation, which is just through the electrical stimulation through the skin, maybe at the area of the ear. And we had a very, very great and interesting discussion about this, but also about what are the mechanisms, you know, that underlie this. And a lot of the mechanisms may be actually associated with what's happening in the body versus what's happening, you know, through the brain. So this nerve is very important in regulating our inflammatory response, so inflammation, and relaying those signals back to the brain. It also plays a very important role in metabolism. It's regulation of metabolism, and we know that many psychiatric disorders have a metabolic dysfunction component to them, either in their pathophysiology or in their treatment. And then this morning we had Professor Quyen talking about the gut microbiome and the connections with the brain. So I just think it's great that we're also moving beyond the brain to think about the connection between the brain and the body and the implications that has for our general health.
Yeah, definitely. Yeah, interesting. Yeah.
And also related to this, I think that now we are putting more emphasis also how environment, and so how other interventions above pharmacological intervention can be helpful for patients. So how there were several symposium and talking about how, for example, exercise, but also, of course, nutrition can, on the top, of course, of antidepressant medication, can improve the symptomatology in patients. So we are giving, I think, more and more importance on, again, on the environment, because we do know the environment can somehow increase our vulnerability to mental disorders, stress, for example, but it is also true that a positive environment can somehow reshape the vulnerability or can push down vulnerability, promoting resilience and also, of course, the response to treatment. So I think it's something that we should work more.
Yeah, really nice to see that also the scope of ECNP is broadening towards these.
Yeah, so it's not just focusing on
only pharmacology,
but also other stuff.
More holistic. Yeah.
And I think the Love Your Brain sessions
really
really highlighted that as well.
The one on art.
So there was one on art and music as well.
Yeah.
And then today we had one on neurogenesis and the impact of, I suppose, nutrition and lifestyle factors on neurogenesis, which is the growth of new neurons in the brain. So it's interesting, and what maybe we can think about to maybe protect our brain and protect, ensure optimal brain health moving through life. So I think the idea of preventative action, as well as you mentioned, with pharmacological interventions, bringing all those elements together at the conference was really insightful and
definitely.
I think interesting.
Yeah. And what are your plans for tomorrow? Any things you're looking forward to?
Well, actually, related to this, there is a campfire session on food and brain, food for brain, actually. So, yeah, I think it will be a great campfire session. Yeah.
Nice.
I have to chair a session on gene therapy, and that's I think gene therapy is getting more and more accepted now that there are also some of these AAV vectors are registered for eye disease. And well, for instance, today Deborah Massini from Stockholm told us that even if you have no dopamine anymore, which is animal experiments, you can still target certain brain areas. And then you can chemogenetically reactivate them, and then these animals start to move again. Whereas without dopamine, of course, you don't move a lot. And that, of course, can be translated because we can use deep brain stimulation. But even chemogenetics, theoretically, there's no reason why we shouldn't inject people with a viral vector in the brain if they really suffer, because it's doable. We have the technology. So I think, you hear more about this tomorrow.
Something for the future.
Well, tomorrow.
Yeah. Tomorrow, very soon future. Yeah. For you, Olivia?
So, unfortunately, I'm going to leave very early to return home. But the wonderful thing about this conference is the ability to be able to watch some of the symposia and the educational sessions and the plenaries online. So when you can't be there. So I'm really looking forward to one in particular, which is focused on human-derived induced pluripotent stem cells. So again, related to plenary that we had today on organoids. So I think that, you know, I'm really looking forward to that. I think it'll be fascinating.
Yeah, and I think you have a couple of months to watch it.
To
even.
actually watch
So,
what
yeah.
we missed. Yeah, exactly.
Yeah. And for you?
I was saying that there is this Campari session tomorrow on food for brain.
Yeah.
So,
Great.
yeah.
Yeah, lovely. Well, so many interesting things we've seen today. Some things yet to come. Tomorrow, last day. Any other tips you have for the audience? Maybe.
I just
some
think.
last.
No, I just think, you know, there's so many wonderful sessions, and I suppose you just have to plan well in advance. That's where the app is absolutely great.
True.
And plan and have your own calendar. And the fantastic thing is if there are some sessions that are overlapping, that you can, you know, view them later and go to the ones that we know are in person, like the campfire sessions, for example,
Yes.
where you won't get an
Yeah.
opportunity to see those online.
And
But,
be there
I can
early at the campfire
exactly,
sessions.
because not only to the campfire session, because I think that the people listen to the advice that was given the previous day, because today I was chairing one session and I arrived 20 minutes earlier, and people were already there to get their seat. So,
That's good. Good to hear.
yeah.
Well, thank you very much for being here, for giving all your advice. And I hope, you two at least have a nice 4th day of the Congress, and you can enjoy it online, hopefully. Yes. And thank you very much for watching.